Hello, my name is Grant Harrison. It's August 15th, 2021. And upon reading the Gene Therapy Comes of Age article by Dunbar, Joung, Kohn, Ozawa, and Sadelain, we are presented with an optimistic outlook on gene therapies. The authors of this article outline the historical context of the methods that have been presented in the past, with certain complications, but also provide insight into the direction that gene editing seems to be going. In addition, the authors provide context to what current and potential future barriers may arise as new technologies and advancements will allow for treatment of many types of diseases. In their historical review of previously studied methods, the authors discussed how initial methods of gene therapies such as replication defective retroviruses, adeno-associated viral vectors, or AAVs, they came with the consequences of therapy-related toxicities, inflammation responses to the vectors, and malignancies due to the activation of the proto-oncogenes. These initial vectors were not evasive enough to avoid causing off-target unintended effects of the therapies. However, lentiviral vectors improved the efficacy, efficiency, and safety of delivery methods, and research of clinical trials were allowed to proceed into the 2000s. Engineering of lentiviral vectors in the late 1990s and early 2000s sprung us into the age of editing via site-specific endonucleases such as zinc finger nucleases, TALENs, and most recently CRISPR-Cas. However, these technologies are under intense investigations to ensure that no unintended off-target mutations result as a consequence. In addition, as gene editing with these may be performed ex vivo using germ cell technology, there arises an ethical concern as human stem cells are a controversial topic. Currently, germline editing is not funded by the U.S. government, and the FDA will not approve clinical trials of that sort as well. These standards are applied globally. The authors conclude that although the future of genetic therapies shows promise, there will need to be an ethical consensus of how germline technologies may be used, as well as how these types of treatments will be paid for and on what basis the treatments will be awarded. My personal opinion on this matter is that I often reflect outside of my studies. In summary, my personal opinion is that a human germ cell or developing embryo should have a place in the development of genetic therapies, even to the point where we will begin to meet people as adults that were genetically modified prior to and during fertilization in clinical trials. Although humans are not an ideal model organism, as the effects of gene editing may not even be realized in a person's lifetime, I think we may be capable of providing these individuals with normal lives without extreme unintended circumstances. Thank you.